So linkage studies, they're currently still done, but they were very popular during the 80s through about 2000. These studies tended to be, well, they didn't tend to be, they were family-based. So you had to have multiple family members for these types of studies. The number of markers tended to be pretty small, very small by today's, by comparison with today, where there usually a panel for linkage was around 400 genetic markers across the genome. These were microsatellites, which tended to be kind of repeats of DNA variants. Candidate gene studies became more popular in the mid-90s. I worked on one for my dissertation work through about 2005, and again, there's still a place for them, but they were really popular during this this period of time. That's where you perform a focused analysis. So you either select a set of genes based on some kind of knowledge about their biology or other criteria, or you may select one gene, and a lot of times you'll just pick a few SNPs out of it, like 3 or 4. I think one of my dissertation papers was based on 4 SNPs in, a candidate gene. Nowadays, we deal with many, many more SNPs, variants. Genome-wide association studies, or GWAS, that really became feasible after 2005. That's where the number of genetic variants really dramatically increased. So now, rather than using a handful of variants and testing them, it's commonly now over 1 million of them that you're doing at a time. Next generation sequencing, which really only became feasible on larger scale after 2010, based on whole exome or whole genome being approximately up to 3 billion sites in the human genome that we're looking at. The tests that we do on these are often very simple tests that we all learned like in our first year of graduate school, linear regression, but a lot of it is the number that you're doing. As the studies have gotten larger with respect to the genomic coverage, the required sample sizes to achieve good power have also increased. And this is because the multiple testing issue primarily. So now you're doing many tests, and you have to account for that for your p-values. So you need larger sample sizes to help overcome that. And what that kind of led to is genetic consortia, where individual studies are really kind of joining up. Next generation sequencing, again, has really become done on a larger scale just in the last, within the last decade, definitely. Just a few years ago, a whole genome cost over $25,000. Right now, it's around $1,000 per individual. The cost is a function of coverage, so you can get varying levels. You can get cheaper sequence panels that don't have as good of coverage, meaning how well are they actually capturing the variants. And of course, higher coverage is more expensive. Challenges in analysis is that once you move to sequencing, you're getting much rarer variants than what, say, GWAS panels would get. And for looking at any of them individually, your statistical power is lower because it's a rare exposure, basically. So one thing that we sometimes do is we try to focus the tests on a subset of variants, such as ones that may a priori be more likely to be functional or to be important. One area of focus would be focusing on just the exome. So the exome is less than 1% of the total DNA. It's the coding regions. It's the exons, the parts that specifically code proteins. It tends to allow for higher coverage, meaning that the sequence data that you're getting is more accurate. The exome variants, again, is thought to potentially be more important than genomic variants as a whole because they're known to code proteins. One thing that we have found in the last few years is that might not be as true as we thought ahead of time because people are finding a lot of things outside of these areas. Some variants within the exons are clearly bad, meaning they have a very specific function that you know of. For example, a nonsense variant will stop the coding of a protein. Also, we often use prediction algorithms. A couple of them are polyphen and SIFT that can help give you information on which variants might be more functionally important. So the exome sequencing project, or ESP, is a project I worked on. It was around 2009, 2010. It was funded by NHLBI. It was part of the Stimulus Act. Originally, it was supposed to be a 2-year project. It was extended to a 3rd formal year, although people continued working on it for years after that. It was to perform high coverage whole exome sequencing of approximately 7,000 individuals from among 20 studies. And 7,000 doesn't sound like that much, but in 2009, exome sequencing, that one started off, I think it was around 12,000 per person to do an exome sequence. It definitely went down over time, but it's still pretty expensive to do that. This study was focused on European Americans and African Americans, and the goals, of course, were to discover new genes and mechanisms related to heart, lung, blood-related phenotypes. And part of it was this was really one of the very first large-scale sequencing studies. So a lot of it was just figuring out how to work with these data. So 18 to 20 cohorts, depending on how you count it. But these were focused on heart and lung disease. As I mentioned, it was whites and blacks. We ended up with around 6,800 whole exomes at the end of it. Samples were sent to 2 different sequencing centers, one of 2 sequencing centers, either the Broad Institute or the University of Washington at Seattle. One thing I didn't mention, I left out the slide of studies, but these are a lot of the large cardiovascular and pulmonary studies were involved, epidemiologic studies like the Framingham Heart Study, the ERIC Study, MESA, Cardia, Jackson Heart Study, COPD Gene. There's several others. So the way they did this study is they basically got investigators from all these different studies together, and the sample sizes across these studies represent much more than around 7,000 samples. I forgot to mention Women's Health Initiative was also included, and that's a very large study in terms of participants. But it was really kind of open-ended. They said, well, we'll give you this money to do the sequencing, and you've got to figure out how to select a subsample of 7,000 from across all these studies. So the way they did it, they formed working groups across different phenotypes, and they kind of, the leadership kind of tried to decide what are some really important heart, lung, and blood phenotypes. And then the working groups each kind of had to make a case for how many of the 7,000 samples that they would get and how the samples were selected. So what they kind of considered the most important one was early MI, so early onset heart attack. And it was really interesting because I was kind of involved in the selection across all of the different groups pretty much. And the NHLBI kind of allotted the most samples to early MI. But it was just an interesting process because each group had to come up with their definition of cases and controls. And for early MI, they came up pretty early with MI. I think it was less than 45 in men and 55 in women. They had certain case control samples that were from the Broad Institute that met that definition, but across these large CVD cohorts, there just weren't that many of them. I think the total sample that they were allotted out of the 7,000 was 1,200. And I think it actually eventually went up to 2,000. So we ended up kind of pushing that age limit a little older. I think it ended up more like men less than 55 and women less than 65. But it was interesting. I was on a call once, and they talked about controls. and how to select the controls for these. And, you know, if you're thinking that you really want population-based estimates, you're going to want your controls, obviously, to be as well matched to your cases as possible. But for these, you know, really rare exposures, these rarer variants, we're thinking more about statistical power. We're thinking about, we want power, so you want the most extreme that you can get. That's the way to maximize your power for a given sample size. And we all think, or a lot of people in genetics think, well, later on we can figure out what the population effect estimates are. So there was this kind of going back and forth where my thought and several other people's thoughts were, let's get the oldest, healthiest people as controls and then compare them to the earliest MIs. Sometimes that, you know, in epidemiology, you're taught that you want them to compare, but again, to maximize your chance of finding an association for a given sample size. This is what a lot of us thought, but other people were really concerned that if you find something, we don't really want it to be a gene associated with, say, LDL or known risk factors. So another kind of the other side of it wanted to match on what they wanted to do is match on Framingham risk score. And this is a score that's based on a lot of CVD risk factors, such as smoking, hypertension, LDL cholesterol. So that's what we ended up doing. I remember like, oh no, this may not be a good idea. But, you know, it's just interesting going through the whole process. I co-led the LDL cholesterol group where we did just pick pure, the highest and the lowest. And we started out, we were allotted 400 samples. So, and we had 2 ethnicities. And one thing with rare variants is the rarer a variant association, the more likely it is to be population specific. So you really kind of have to do ethnic specific analyses for rare variants, at least as a one stage of your analysis. So for our 400, we picked 100 the highest, 100 the lowest in the whites, and the same for the African Americans. And that was interesting, too, because, again, we had up to 20 studies. They didn't all have LDL measured, but probably there were more than half of them. I'd say about 14 of them had LDL measured, and you're trying to pick your most extreme LDLs out of those 14 data sets. And you can't just pull them all together and pick your most extreme because, obviously, there's a lot of potential confounders in there. So there was a lot of calculating residuals and trying to account for this in different ways. And then also, we generally tried to match within cohort. So if you picked an extreme high in a cohort, you also wanted to get an extreme low from them. The BMI group did very high, but they chose to do more normal as their comparison. And then so on and so forth with these different definitions. One thing to keep in mind is that at the end, a lot of times, even though your phenotype of interest may have only been, you know, selected for in a subset, for example, LDL cholesterol, originally 400, and then it was 600 out of the total 7,000 were selected on LDL. When we analyzed it, a lot of times we wanted to use everybody in the sample that had LDL. So a lot of these samples, even though they were selected for, say, asthma, they had measured LDL. So talk about a non-random sample. This is like this sample that's pieced together from all these very highly selected subsets. So that made for some really interesting analytic challenges in this. So to perform the association analysis, we did just like GWAS, where we just looked at one SNP at a time, and a SNP basically has 3 different genotypes. A SNP meaning single nucleotide polymorphism, has 2 different alleles, and usually 3 different genotypes. So a very simple analysis. We can also often just treat it like a linear regression for a quantitative trait, a logistic regression for a case control. The thing is, in this setting, is that a lot of these individual variants will really be too rare. Even with 7,000, you're just not going to have the power to detect associations with individual variants. But one thing that we can kind of take into account, or that can potentially help us, is often once a gene has one important rare variant, there's often more than one, meaning there's often can be multiple rare variants. This is known in certain genes. For example, the Ldl receptor, it's known from Mendelian studies to be associated with, it causes familial hypercholesterolemia, and basically, there are many, many, many individual variants that are cataloged for that. So we kind of take advantage of this fact, and we also utilize in this setting gene burden tests. They're called different things. Sometimes they're called rare variant aggregation tests, gene burden tests, basically where now you are collapsing information within the gene rather than just the variant being your unit of measurement. It's the whole gene. So you collapse information from the gene into a single test statistic, and now that's your predictor. So again, the idea is to aggregate information across all the rare variants that are in some defined region. It doesn't have to be a gene, but it often is a gene. For example, the exomes in a given gene. And then you use that aggregate data as a predictor. So the most simple one, and the one that was really used a lot early on, where, well, this one says sum them, but a lot of times you would just, you would start out looking at the minor or low frequency of every variant in your sample. And then you define something as rare, like maybe 1% minor or low frequency in your sample. Now you look at the coding regions in a gene, and you go to each variant that is now has a minor or low frequency less than 1%. Any of the ones with minor or low frequencies greater than that, you just kind of ignore them. But any of the ones that are rare, you look at the person. Do they have the common variant, the common homozygote, common variant homozygote, or do they have at least one copy of the rare variant? If they have at least one copy of that rare variant, you code them as a 1, meaning you're not counting them as exposed. So for example, somebody might have a variant at 2 different rare variants, the variant allele at 2 different rare variants. They would still just get a 1 even though they have 2 of the variants. Another way would be that you actually add up the rare variants. So again, now it's just a very simple collapsing. You can also define this in different ways, like maybe all potentially damaging variants that you focus this on. You can change your minor or low frequency. And the why minor or low frequency, you might think, like, why would rarer be, something you want to focus on? But there is the idea, and there's evidence for this, that the rarer a variant is, the more likely it is to do something bad. Because if it is more common, then it's not doing anything too bad, so it's not getting selected out. So the idea is the rarer it is, the more likely it is to be bad. So it's a good set to focus on. So there's many forms of these. A lot of times, the CMC that I mentioned here, combined multivariate collapsing test, that's the simple one where the person either has it or they don't have a rare variant, or they don't in that gene. A lot of times we use that, and I'll show you, we define them slightly different based a lot of times on their minor or low frequency. But there are minuses to this approach too. For example, what if you have a single gene and there might be some variants in that gene that, say, increase your mean level of a phenotype, a continuous phenotype, and others that decrease? This approach isn't going to work well, right? Because it's just literally counting you as one or the other. So the Scat test, sequence kernel association test, is what was developed, and it's pretty much very widely used now. It will allow for positive and negative effects in a gene. People also call it gain of function and loss of function. It's a type of gene burden test, but here you don't actually start with the SNPs and categorizing them. Here, each SNP is tested individually, like you do individual SNP tests, and the results are aggregated after the test is run. So again, the plus here is that it allows some to be positively associated and others to be negatively associated. Now, if you do have a gene where all the variants in it are positively associated, for example, then the Scat test isn't going to be as powerful as if you counted them all in the same direction. But in general, without knowing ahead of time what the mechanism is, this is a pretty good test. And the Scat-O is just another version of the Scat, which is supposed to help it come a little closer to if the situation is where they're all in the positive direction or all in the negative direction. It gives you a little bit power, a little bit more equal to if you had just done the simple test. So these rare variant aggregation tests, they're not that complicated to run, but what we really found really early in ESP is that just slight changes in these can have humongous impacts. In fact, when we were first running these, we would just make a slight change to the definition of which variants to include in the aggregation test, and we would just get completely different results. Our top genes, like in the top 20, would just be completely different every time. So that was very frustrating, and as you can probably guess, that meant we didn't have great power. But this is an area that's still really an important part of methodology development. How do we figure out which variants to include in these burden tests? So again, some of the approaches, again, minor low frequency is a big one. Predicted function is again one that's used. I mentioned these that there are databases that can help you understand or give you an estimate of predictive function, but also just focusing on the small set of variants that are more obviously functional, which we often call them loss of function, which would include nonsense, frame shift indels, read-through splice errors. But again, okay, so this is mostly what I've been talking about is for exome sequencing, where you have a natural unit of what you can collapse in. The gene for a gene, an exon, it's pretty well defined from your data, from exome sequence data, that you know what were in exons, and you can say, I'm just going to include the variants in those exons. When we move across the whole genome, then this becomes so much more challenging that people don't really know how to define what the blocks are now. A lot of people now are just, you can use a moving window where you start and you just march along. But again, once you move to whole genome sequence, it's a much bigger challenge. Another thing that was a big challenge in ESP, and I worked very closely on, was phenotype harmonization. I mentioned there are around 20 studies involved, and there can be a lot of differences for those of you that have worked on different studies, just the way they name variables, the way things are measured. There can be a lot of differences across the studies. And there can be many, many reasons for this. For example, the Framingham cohorts, or the Framingham heart study has 3 different sub-cohorts within the main study, where they have the original cohort, then they have the offspring cohort, and then they have a generation 3. And they keep everything in separate data sets. They call things different variable names, and then there's multiple visits. So the Framingham heart study, literally, I think I had a collection of all their variables, and it was over 30,000 variables that they have across all these different data sets. So I was in charge of actually harmonizing the phenotypes across these studies, and got to spend a lot of time just looking at lists of variables. So this is just a list of a small selection of hypertension-related variables from ERIC Cardiovascular Health Study, Cleveland Family Study, Framingham, Jackson Heart Study, and MESA. And you can see just something as simple as hypertension, that there's many different names for them. There's different classifications, there's different visits. So kind of putting that all into one data set was quite a job.
Can I?
Yeah, please do.
I can't ask questions about the genetic parts. I'm going to ask questions about
Yeah.
So did you, when you harmonized the phenotypes, Did you use any of the current, tools?
Right, like Phoenix?
Like OMOP, for example?
Yeah. So
we're doing something similar with data from the search registry.
Yes.
And a similar registry of diabetes in children in India. And it takes somebody like Michael Kahn
Yes.
to organize.
Yes, and I'm working now with Compass a little bit and learn. Yeah.
And
so
we harmonized to an external.
That's right.
Nomenclature.
Yeah. So that's always the gold standard. If you have the ability to standard, to these like, OMOP and
Phoenix.
The Odyssey Phoenix, yeah. So I spent a lot of time with Phoenix people and trying to do that. The issue there was for a lot of these studies, data were collected sometimes years ago, sometimes decades ago. So you couldn't, in some cases, standardize to what, Phoenix at least has very specific definitions and very specific measurements. If you're starting a new study, it's awesome to do that. But the problem is, it would say, blood pressure has to be measured this way. It has to be an average of 3 different measurements or something like that. And a lot of times studies wouldn't have those definitions. So a lot of times we would have multiple layers. We would have maybe a tighter definition, and we would try to use the nomenclature as much as possible. But then, so we would kind of have a lowest common denominator, meaning what can be the most harmonized, and we would have that, and then we would have a more, maybe the more ideal or maybe more consistent with some of the standardizations. So there's limitations, and when I say harmonization, that there are things you can't completely harmonize, right? That are just never going to be truly harmonized, but you do the best that you can. Luckily, I'm glad that these things exist now. So as we move forward in studies, that there will be less the need for this. So additional challenges that occurred in ESP, again, I kind of have mentioned this, but there were many different criteria for sample selection across these studies, and I was also involved in the sample selection. And a lot of times there were multiple of these going on at a time, so there would be the early MI group, they want to select samples across these 20 studies, and then the LDL was selecting. Well, you might have already selected somebody for early MI, but we wanted that same person for LDL, and they were kind of happening at the same time. So it was kind of a crazy time trying to keep up with it all. Batch effects were a major issue in ESP. Batch effects, I'm guessing most people here, but know that this can be a major area of confounding. For example, here's just a few. One obviously being people came from different studies, for example, but the samples were sequenced at 2 different study sites. And epidemiology 101, what you would like is that was randomized. So who was selected for who, and from each study that this would have been randomized where they were sequenced. No, in some cases, all the cases were sequenced at this site and all the controls were sequenced at this site. And I remember just like losing my mind early on and saying, that's going to be awful. And this was a $60 million project too. So it's like, you can't really easily go back. And people tell me, there's not going to be batch effects. It's going to be fine. And then I remember, I'll never forget seeing the first QQ plot for early MI. And it almost went off the page. It was so crazy, because all of the cases were done in one set and all the controls in another. So there were things that had to go back and get redone to some extent. Some of it was supposed there was a little bit maybe a black box going on at the University of Michigan where they did a lot of filtering. I think we lost a lot of variants because of that. But batch effects were a major thing. And even just within sequencing site, the sequence machines changed over the time of the study. And again, everything was done in batched. It was done batched by phenotype group. So lesson learned going into the future. Just identifying optimal QC, usually like when it's not sequencing, I know how to go to a study and I know what to look for and what QC to do. We really didn't have any idea early on with this. And also, figuring out the analytic approaches. I think I mentioned the the gene burden test, but just trying to figure that out. Replication challenges. We kind of had already taken all the biggest heart and lung studies, so where do we go to replicate? That was a big challenge. So I'm just going to show a couple of slides from the LDL paper that came out of this. I was a little bit proud during the study because ours was the first study to actually get a significant p-value. Now, that was the upside. The downside a little bit was these were mostly known LDL genes, but still, at least it showed our data was reasonable. Background for LDL is that there are known monogenic conditions that lead to very high or low LDL. So Mendelian linkage studies have been done and have identified a certain number of genes. There have been very large-scale GWAS for LDL. I think it's around 56, although I don't know if that's the latest iteration. Explains about 30 to 40% of the heritability, which the heritability is the variability of a trait, of a quantitative trait due to genetics. So there's still, there's this whole term about missing heritability is maybe the rare variants account for some of the heritability that hasn't been explained by GWAS, which largely cover more common variants. So we use linear regression for the continuous LDL, and again, this was in all of the samples combined, anybody that had LDL measured. So say you were selected for low BMI or average BMI, and you had LDL, we would still analyze it. As you can imagine, there was a lot of iteration in how to deal with the selection of those samples. In some cases, when you put together a bunch of really highly selected samples, you can get increased type 1 error just from that. And then we used logistic regression for kind of the focused analysis for just the ones that were selected for our phenotype. Just a typical covariate adjustment. We adjusted for what group they were selected for. And we also accounted for PCs for ancestry. Lipid-lowering medication for LDL, we did not adjust as a covariate. We actually adjusted for that before we did the analysis because it's been shown to treat as a covariate can cause some issues. So for the single variant tests, so we performed single variant tests, and I can't remember, we did have a lower limit for minor allele count. I think it was like at least 10 copies of the variant in the whole sample. For gene burden, what these numbers are, these are that really simple test, and the numbers there just are the different minor allele frequencies. So T5 would be anything, the burden test focused on genes where you're only including variants with less than a 5% minor allele frequency. T.1 would be less than 0.1 minor allele frequency. So the T.1 is looking for the rarest variants. So we did it based on all, all non-synonymous variants. Sorry. I'm just getting over a cold too, so my voice is maybe losing it a little bit. All non-synonymous, so those would be or focused in the exomes. And then that caused an amino acid change, and loss of function variants, which is a subset of that, which are kind of the worst of the potentially worst. So we did find significant evidence for TOM40. So TOM40 is a gene that's actually next to ApoE, and ApoE is a known associated, the haplotype in ApoE is known to be associated with LDL. The ApoE haplotype itself wasn't well covered in this particular, apparently the haplotype's very hard to measure, but this TOM40 variant tags the haplotype very well. And additionally were these four genes, PCSK9, Ldl receptor, ApoB. These are all well-known Mendelian genes. We also had suggestive evidence for 2 additional ones in the gene burden test, and these are also known LDL genes. So at least we had kind of a nice proof of principle. People were at least happy in that, the data at least were finding known genes. And it was interesting in that those genes had only been identified previously in linkage tests, so where you collected family members that had a clinical diagnosis of hypercholesterolemia. Here we were taking population-based studies and doing a very different kind of test, so it was reassuring. Yeah.
So I wanted to ask about
Yeah.
that. What was the purpose?
What was the purpose of doing this?
Yeah.
It's a great question. 60 million?
Find the gene that we already knew
Yeah,
about,
so the
or-
idea was that we would find new ones. The one PMPLA5 actually was not a previously known LDL gene. So that one, we did see an association. What was kind of nice about it was that the mechanism of it, it was only the gene burden. It was only the really rare. So it was a good reason why you would not have seen it in a GWAS. PMPLA5 was the novel one. It barely met significance, and so we wanted replication with it, right? That's really the only way you're going to know. And the problem was there really wasn't many places to turn for replication. We didn't have another 10 million sitting around to get some more extremes to sequence them. But there was one study, it's a diabetes study, T2D genes, that was also doing sequencing and had LDL in a subset. So we were able to go to them, and the p-value was like 0.04. So barely significant again. And I will tell you, this was from like 2014. I haven't seen it replicated since then, but there's not a ton of sequence data out there. So we're still kind of waiting to see if this is a real result. But I think the big story was they vastly overestimated what they thought the effect sizes were going to be for these things. They thought it would take smaller sample size, and it became really clear. Not a huge number of papers came out of ESP. So there's been a few, but really goes to show you need big sample sizes. Yeah, were you going to say? Yeah.
In the
Go back.
In PCSK9 or the known
loci,
if you changed your minor allele count for your gene-based test, could you do some QC on like how best to structure your gene-based
the burden test?
Yeah.
Yeah. So the problem with that is, you could certainly go back and for any particular sample say which were the variants I should have, or you could maybe look at them individually. And the problem is, how do you assess significance at that point, because you're completely snooping. Now apparently Shihong Lin at Harvard is working on this. In fact, she's just requested, I'm now working in whole genome sequencing data through the Top Med Consortium, and she's requested the phenotype I'm working on there, BMI, to use as a test case. So I'm going to, I think, be learning a bit more about that.
I just wonder, like, if you go down to 2 count, a variant that's only represented twice, if you just exclude singletons, like if it helps you at all.
Yeah. I think a lot of us think about that. We wish we could go in, and you could imagine an ideal situation is you go through each gene and you look at all the potential combinations, pick out which is the best one, and account for the fact that you looked at all the combinations. But then you would have a replication sample that you could replicate it. I think that's the ideal situation, or close as you can get. But I don't know exactly why that's not being done. I guess part of it is we still are just trying to get the biggest sample sizes for discovery, so nobody has the replication part of that. And I know I'm way behind, but I've got this in pieces, so if I don't get through all of it, it's fine. But this is just to kind of show you a little bit of differences in ethnicities. So what this is just focused on the extremes, meaning you were selected either for a very high LDL or a very low LDL. And that's whether you're above the horizontal line or below. There's 2 of them. The one on the box on the top is for African Americans. The box on the bottom is for European Americans. So, and then the length of the vertical line is how many copies of the rare variant you have. So, as you can see, most of the action is happening in the African Americans. There's hardly anything going on in the whites, which is at the bottom. But the top, you could kind of, like, if you pictured putting that up on its side, which way would it tip? And The African American one, you can tell it's definitely going towards lower LDL because there's more line below the vertical or below the horizontal line. So rare variants, and it was known ahead of time that in African Americans that there were two rare variants associated with quite a bit lower LDL. In fact, there's a medication now. There's a class of medications, PCSK9 inhibitors now, that have been kind of designed off of PCSK9. But as you can see, not a whole lot happening in whites there. And this is a similar plot for LDL receptor, where in general, LDL receptor variants are almost always seen in people with very high LDL cholesterol. One interesting thing in this that happened was that we saw one person that had a really low LDL receptor variant, or low LDL cholesterol that had a variant in the exome. And I'm not sure that that's really even been well reported. But kind of similar action happening in whites and blacks. Again, there's more, it looks like a stronger effect in the whites, which is at the bottom. But just to give you kind of a sense of the kinds of things that we look at. And you can see the vertical lines are all kind of the same height, meaning they all had one variant except that one over on the right on the top. There were two for that one. And then this is just to briefly show you different kinds of burden tests. So the different columns are different burden tests that we ran, and then the rows are different known genes. And the ones that are highlighted are ones that the darker pink were ones that were actually significant. So it just kind of goes to show you that different burden tests work better for different kinds of genes. Most of them worked pretty well for PCSK9, but for LDL receptor, it was really only the ones that either focused only on the loss of function variants, meaning the really bad ones, or the ones with really rare variants. And then ApoB, it was really only the loss of function that would detect that. And then ApoE, which is really tagging this was the one that was the Tom40 variant, is just a single variant. But it kind of goes to show you can see we did some Scat there, but the Scat it didn't necessarily, for example, for LDL receptor, the Scat test was not significant at all. But it goes to show that, we may need to be using different burden tests to get different genes that have different genetic architecture.
So how do you know which one's the right one?
That's the big question. Nobody knows, and it's so frustrating because you'd like to do, and this is just a small number of the possible ones you could do. Ideally, you'd want to do many of them, but the sample sizes would just be so big to try to account for correcting for all those different ones you do. So in some sense, it's maddening. It's a maddening process. So I'm going to talk a little bit about the TopMed consortium. So this is moving into whole genome sequence now. So again, it's NHLBI, and traits here are heart, lung, blood, and sleep. So this is not only just focused on whole genome sequencing. So now we're moving from whole exome, which is like 1% of the genome, to the whole genome, like the 3 billion sites. And but it's also incorporating other omics, such as metabolomic, proteomics, transcriptomics, etc. So more studies here. There's over 30 currently, maybe potentially a lot more than 30. I haven't added recently. Pretty much every year for the last 4 years, they add another layer of studies. They're on their 5th one now, I think, and then there's one more planned. So this one doesn't just have whites and blacks, but also has Hispanics, Latinos, and some other subgroups, such as Pacific Islanders. So this has more sequencing centers, so not just 2, there's more of them. There's an informatics research site at the University of Michigan where they're harmonizing the sequence data. So they kind of call the data all together. So to detect the variants, the more people you have to detect these variants and being called together, the better. So that's what they're doing. And then the University of Washington is leading the phenotype harmonization activities. So there's data sharing through dbGaP exchange area. And these data are being released very quickly into dbGaP for public use. So they're released pretty quickly where pretty much anybody in the scientific community can request the data. This is just a very quick, I don't expect you to see really any individual, but a listing of phase 1 and phase 2 cohorts. We're now up through phase 4, so you can see lots of studies. And they're currently picking phase 5 studies. I'm glad I don't have to harmonize all this, to be honest with you. Okay, so here it's about 30x coverage. So just to quickly tell you what 30x coverage means, is when you're measuring it, it's the x coverage is the number of times you're actually seeing each variant when it's measured. So the more times you see it, the more times the more sure you are that is a variant. For example, 5x coverage, you're only seeing that variant in a sample like 5 times. So the more you see it, the more sure it's truly a variant. I don't really need to go through all of these details. So I'm an investigator with the Jackson Heart Study. They were one of the first studies to get sequencing done as part of this. So this just gives a little bit of the QC that we did on it, where we identified a couple of pairs of monozygotic twins where one hadn't previously been reported in the pedigree file. We check concordance with dbGaP fingerprints, which is usually like a small GWAS panel. And then we found some sample mismatches, which is very typical of this. And we were able to resolve about half of those we were able to keep, and half of them we had to discard. We exclude samples that are based on high levels of missingness. And then we also exclude variants that are monomorphic, meaning there's no variation in our sample, because you can imagine this takes up a lot of storage space. So we try to, there's different levels of these files that you get, where the smallest just being only the variants that have a variant in at least one person. So TopMed BMI were one of the first to get some analyses done as part of TopMed. And just very briefly, I'm going to go through it. This is just, here rows are some of the samples, the cohorts that are involved, and then the columns are different ethnic groups. And it just kind of shows you that some of the cells have pretty big numbers, some of them, very small. But again, I'll readdress that in a slide or two about why that's an issue. This is just BMI and height. I said just BMI, but this is actually BMI and height. So the top here is distribution of BMI over what we've looked at so far. You can see the third one in. There were some issues with BMI and going back. Turns out they had their height and their weight swapped. But these are things that happen. I mean, they happen even when you're doing these crazy expensive studies. You have to check these things, And then height's on the bottom, more reasonable. There's some reasons why for some of those outliers. Preliminary analysis so far. So one thing we're doing is, so when you're really looking at these rare variants, or just when you're looking at these small p-values for GWAS settings, regardless, you're more, you have to worry more about the distribution of your assumption of normality. Okay, so when you're looking at for a p-value less than 0.05, you can get away from if your distribution isn't really normal, it's not such a big deal. But when you're looking for p-values of 5 times 10 to the negative 8, then departures from your normality become more of an issue. So one thing that's done, and particularly with rare variants, one thing that's done a lot in these settings is we inverse normalize them. So basically you just take your continuous trait, you rank it, and then you put that onto a normal distribution. And the issue, one of the big issues that we're dealing is we're trying to do this within studies, sex, and ancestry. But you saw that table. There's some cells that have very small numbers. So we're working on collapsing those, hopefully in an optimal way. So far we've excluded age less than 18. There's a few samples that have that. And there's some selected samples for certain diseases that we thought might be too associated with BMI to include, so we excluded those. Okay, so pooled analysis where we include the blacks, the whites, and Hispanics, where we looked at the single variants. We also did it stratified for whites and blacks for the single variants. And then we have done some of the Scat tests as well. There's a line cut off, but I don't think it's that important. But for sample size, there's around 9,500 of the blacks, and there's around 25,000 of the whites. I don't really need to go into this, but basically these are the QQ plots, and it's basically that they're pretty reasonable, meaning there's no big departure from or thought there's that we're not accounting for say hidden relatedness or something. The height one is on the right. That one departs a little bit early, so maybe there's a little bit of a problem, but we don't think anything too major. Just briefly, these are Manhattan plots for BMI and height. Excuse me. And if you know BMI genetics at all, FTO is a well-known locus, and that one shows up, but not a whole lot there for having 25,000. So it goes back to like, what are we doing here? But for height, we have more there. We're also maybe halfway into the sample size, so we're going to be able to double our sample size. But still, for the single variant test, the other thing to know is it's 25,000 in whites and a total sample size of around 35,000. That sounds great. But there's been GWASs. I've got to tie it up here soon. I'm almost done. But there's been GWASs where they've done imputation, where they impute the rare variants. And those have been done, I think the current setting is in 1 million people. So they can impute pretty small. So it's not going to be too surprising that we don't find a whole lot new. But anyway, so this is our findings, but overlap with previously known. Excuse me. So basically what it shows here is most of these were actually previously known. So that you can ignore the one on the bottom, but the one on the top is if you see green, that means it's previously known. If you see black dots without green that are above the red line, that suggests something potentially novel. So we might have a couple there, but I'm not going to get too excited until we fix our analytic strategy. Yeah, okay, yeah, let me just see if there's anything. I'm just going to quit there. If anybody wanted to see a little bit of the rest of it, then I'm happy to share my slides. But thank you very much. I went much slower than I usually do through these. Thanks.
Well, just a quick question. Mm-hmm. What are the next steps?
Oh, so next steps, basically we're just revising our analytic strategy a little bit. Shree's question was great because we're trying to work with people doing statistical methodologies to try to improve our burden test, because that's the challenge. We don't know how to do them in this. Pretty much. And gain more sample size.
Thank you.
Thanks.
Our next lead seminar talk will be on May 21st, also in this room with Michaela Palombo.